If you've ever watched crime dramas or documentaries where they use a DNA fingerprint to identify criminals, you may have seen these photographic plates. But what do those seemingly random series of lines on the photographic plate mean? Let's find out more, shall we? Before we start though, we need to understand a little bit about how DNA works. A DNA molecule consists of a big long chain of smaller molecules called nucleotides. Each nucleotide contains a section that can be one of 4 different, even smaller subunits. Each of these is denoted by the letters A, T, C, and G. This means that DNA is a big long string of these letters in specific sequences. The order of these letters is a set of instructions telling our cells how to build proteins, which is the key to life. In actual fact, each molecule is made up of 2 of these strings opposite each other, but I'm only interested in one of them at the moment. This big long string that makes up the molecule is folded up in the chromosomes. Each human cell has an identical copy of each of these chromosomes, and in humans there are normally 46 of these chromosomes residing in the nucleus of each cell. DNA fingerprinting was developed in the 1980s by Sir Alec Jeffreys working at Leicester University in the UK. The original technique was based on something called restriction fragment length polymorphism, or RFLP analysis. This technique analyses the differences between the DNA from different people. Technology doesn't stand still though, and DNA fingerprinting now uses a technique called STR analysis. STR stands for short tandem repeats. This uses a different technique but still looks for the differences between our DNA. I'm going to look at both methods in this video. RFLP analysis uses restriction enzymes. These are enzymes used by bacteria to protect themselves against viral infections. If you haven't seen my video on bacteriophage, then I introduce restriction enzymes in that video. These enzymes cut DNA into fragments, but they don't cut it randomly. They cut it at very specific, usually palindromic sequences. These are called restriction sites. My DNA and your DNA is very similar, but it isn't identical. Most of our DNA is non-coding. This used to be called junk DNA, but we now know that some of it does play a role, so it isn't all completely junk. This non-coding DNA is slightly different between different people. If I take a sample of my DNA and your DNA, then because of those differences, those restriction sites will be in different positions. If I now add a restriction enzyme to both samples of DNA, those 2 samples will be cut into a different number of fragments of different lengths. So far so good, but I can't see the DNA, so currently I just have 2 test tubes of liquid that look very similar to each other. So what next? We now add both samples to little wells cut in an agarose gel. This gel has a similar consistency to the jelly or jello that you can make at home, but it's a bit less strawberry and a bit more sciencey. After I've added the DNA samples to the little holes cut in the gel, I'm now going to apply an electrical current to the gel. DNA has a bit of a negative charge and so will move towards the positive side of that gel. Smaller fragments of DNA can move more quickly and so will move further through the gel. The larger fragments can't move as far, and this process is called gel electrophoresis. All well and good, I still can't see the DNA. Now I use a nylon mesh and press it onto the gel. The DNA is then transferred to the nylon and dries onto it. This means that the DNA is now fixed in place and won't move around. I can now use short sections of DNA that I now know to be complementary to specific sequences. I'm also going to make these pieces of DNA radioactive. If I add this DNA to the nylon mesh, the radioactive DNA will join to the DNA from my samples in the locations where they finished up after the electrophoresis. So now I have a nylon mesh with radioactive samples of DNA, but I still can't see the DNA, radioactive or not. Finally, I now put my nylon mesh onto a photographic plate. Radiation has the same effect on a photographic plate as light does. it lightens the plate, and so as a result I end up with a photographic plate with the sequences of bands corresponding to those fragments of DNA. Only samples of DNA from the same person will have been cut in exactly the same places and will therefore have the same pattern of bands. So my DNA and your DNA will have different patterns of bands on the photographic plate. This method is all well and good if I have fresh samples of DNA. However, over time, DNA degrades and breaks down into fragments. This means that if I have an old sample of DNA, maybe from an old crime scene, or maybe from a skeleton found under a car park in Leicester that may or may not have been an English king, we have a problem. The DNA has already been cut into fragments by time, and so this technique is pretty useless. Just before we look at this other method of DNA fingerprinting, I make frequent trips into outer space, inner space and through time. If you fancy joining me on my journeys, then don't forget to subscribe. My time and space machine is bigger on the inside, so there's plenty of room for everyone. We are finding out new things about DNA all the time. One of the discoveries is that our DNA contains short repeating sequences, the STRs that I mentioned at the start of the video. These STRs consist of sequences of either 3, 4 or 5 bases repeated a variable number of times in a line with no other bases in between. These STRs occur in a number of our chromosomes. I inherit 23 pairs of chromosomes. In each pair, I inherit one from my mum and one from my dad. These two chromosomes will be slightly different because they came from different people. Let's say for instance that the chromosome I got from my mum has 4 STRs, and in the chromosome I got from my mum, the STRs are 4, 7, 12 and 15 repeats in length. The copy of the chromosome that I got from my dad has the same 4 STRs, but they are 5, 11, 18 and 19 repeats in length. Interestingly, even though the STRs in different people differ in length, the sequence of bases directly before and after the STRs is always the same in different people. We can then use another technique called PCR. This can be used to make many copies of just that repeated sequence of DNA that we're interested in. I can then perform the same electrophoresis technique to separate the different STRs as I did before based on their size. This means that I'm only comparing the length of the STRs, and so it doesn't matter how fragmented and broken up the rest of the DNA is. As long as the STRs are intact, this method will work, and I can identify the sources of old DNA samples. DNA technology is constantly improving, and we're learning more and more about the intricacies and complexities of this most fascinating of molecules. And until next time we explore this fascinating universe that we live in, thank you for watching.